Today in biology class we're going to be doing a DNA extraction lab, and I'm going to be using strawberries, but you can use any fruit. Softer is better because it's hard to smush it up otherwise. Here's the actual lab that we're doing. And here are the questions that they're going to have to answer at the end. So the supplies that we need are strawberries, cheesecloth, some type of bag that closes. It's best to use the freezer type because they're thicker and stronger. You're going to need some pipettes for them to do their solutions. I made up a detergent solution, which I'll give you the recipe for. Here's the recipe for the detergent solution. I cut this in half because I don't need, it only takes a little bit for each group, and I don't need like a whole liter of this stuff. So what I showed you was a 500 milliliter of water, 25 milliliters of soap, and 1 teaspoon of salt. And you also need ice cold 95% ethanol. I've got mine over in the freezer, so I can't show it to you right this minute, but I'll get it out when the kids are doing their lab. So right now I have my strawberries sitting out at room temperature. They were frozen this morning, still frozen. I forgot to put them in the fridge yesterday, but it's good to put them in the fridge the day before, and that way they're nice and soft and smushy to do the lab. At each lab station they have a little test tube rack, and although it says differently in the lab instructions, I'm going to have them use a test tube, and they're going to use tweezers to try to get the DNA out. Sometimes I use beakers because it's much easier to get the DNA out with a beaker, because it forms in the interface between the water layer and the ethanol layer. However, when you've got your funnels and you're trying to drip it down into something, it's much easier to do into this. I may do a combination and have them extract into a test tube and then pour it into a small beaker and add their ethanol. That might be the best solution, and then it'll be much easier because it's really hard to get these tweezers and actually tweeze with them. They only go like this deep. It'd still be able to be functional, and my layer is probably going to be down there somewhere. If you don't have beakers and test tubes and test tube racks, those kinds of things, you can easily do this just into like a cup. You just have to hold the cup so it doesn't tip over, and then you'll have the same result. Another variation is you can have them use a glass stirring rod to get their DNA and try to twist this and spool the DNA onto the glass stirring rod. Sometimes that's fun to try too. All right, step one. We smush our strawberry for 2 minutes. They won't know whose hands are whose. Smushed one?
Oh, I'm famous. I'm on the internet.
All right, you will. Our next step is to open the bag and add 2 dropperfuls of our soap solution.
Strawberry looks slimy. It's smushy.
Yeah.
Is that about full?
That's about as full as you're going to get it.
All right.
That's why I said add 2. Perfect. Now ziplock it, and you guys smoosh it around for another minute or two. Okay, so there's our apparatus, and you're gonna squeeze it in. Perfect. See how it goes down and starts dripping into the tube? And then you can just throw away the baggie.
Who's to cut it? Oh, yeah, that's why.
And there's our solution. And it's dripping down into the test tube. Going. I put it in the freezer in between classes.
I was on the one who study all first day, and I was like talking, so I was like this, and they were just
Just to be.
Oh my gosh! That's huge!
Dropping, bro.
That's a huge glob of DNA.